Hello, I'm Selene Ingusci, and I'm currently a research instructor at the University of Pittsburgh. And today I'm very pleased to discuss gene therapy platforms, and in particular, vector platforms based on herpes simplex virus type 1. So the objectives of today's presentation are to discuss the advantages and disadvantages of gene therapy vector-based platforms. We will look deeper into describing HSV-based gene therapy vectors, and then we will take a look on how region of non-coding DNA play a pivotal role in maintaining transgene expression. But first of all, what is gene therapy? Gene therapy is defined as the treatment of disease by transfer of genetic material into cells to prevent, treat, and potentially cure a disease. And it acts by several mechanisms. Here I'm reporting a few. So we can have gene replacement strategies in which a disease-causing gene is replaced with a healthy copy of the gene. An application of this is, for example, the X-linked recessive Duchenne muscular dystrophy, where the delivery of a functional copy of dystrophin can ameliorate the disease. Another example is gene knockdown. So it's inactivating a disease-causing gene that is not functioning properly. An application of this will be in a dominant disease, where actually the expression of the genes, of the gene product is deleterious. So the only solution we have is to inactivate the gene, and can be coupled with gene replacement strategies. And a last example is gene addition, that is introducing a new or modified gene into the body to help to treat a disease. An application of this is, for example, the delivery of a gene responsible for clearance of beta amyloid plaques in the context, for example, of Alzheimer's disease. But how do we deliver all this genetic material to the cell? There are two main platforms for gene delivery: non-viral vector and viral vector. Today we will focus our attention on viral vectors, since viruses have evolved a natural ability to deliver their genetic material to the cells. So if we are able to remove the genes causing the infectious disease, these viruses become vectors or carrier for our transgene payload. And what are the requirements for a good transfer vector? So it has to show high transduction efficiency for the target cell, high payload capacity to accommodate large genomic DNAs. It should be safe and immunologically inert. It should not cause any insertional mutagenesis, and should provide sustained expression of the transgene. So we will discuss two viral vector platform, one based on adeno-associated vectors and one based on herpes simplex vectors. Adeno-associated vectors, or AV vectors, are the most widely used viral vector platform. They are small, single-stranded DNA viruses with a very limited payload capacity of about 4.7 kb. And therefore, they are very easily to manipulate. The main advantages are that they are not pathogenic and cause a few events of insertional mutagenesis. So here on the right, I'm showing the wild type genome of the AV virus with the viral genes, rep and cap, surrounded by the inverted terminal repeats, these white boxes. So the first generation of AAV vectors were developed by substitution of the viral genes with the transgene cassette, flanked by the inverted repeat. These vectors, unfortunately, showed delay in transgene expression due to the fact that a second strand of DNA synthesis is needed to start transcription. To overcome this issue, a second generation of AAV vector has been developed called self-complementary, where a mutated inverted terminal repeat is placed between two copies of the transgene sequence, one inverted and complementary to the other. This allows for the genome to self-anneal, skipping the host-mediated second strand synthesis, and therefore reducing the time between the infection of the cells and expression of the transgene. Unfortunately, this reduces also the payload capacity from of these vectors from 4.7 to 2.3. So what about herpes simplex vectors? Herpes simplex vector shows multiple features that make them an ideal candidate for gene therapy application. First of all, their natural tropism for neurons, the high transduction efficiency for dividing and non-dividing cells, and their exceptional payload capacity that varies between 50 and 150 kb based on the various HSV vectors that are currently available. Moreover, the vectors is always maintained as non-integrating circular episomes. On the right, I'm showing the wild-type HSV genome in A that is divided into segments, the unique long and the unique short, separated by the reiterated joint region. And I'm also showing the genes called immediate early genes, especially ICP4 and ICP27, that are required for virus replication. So the first generation of HSV vectors has been developed by deletion of ICP4, ICP27, and optionally ICP22. This vector showed a moderate toxicity and unfortunately showed also a transient expression of the payload. So in the following slide, we will go through all the new engineering that allowed us to create a safe, non-toxic, replication effective HSV vector that will allow for prolonged expression of the transgene. So how do we create a safe HSV vector? So transcription of HSV genes is sequentially regulated. We have three distinct kinetic class of genes here reported as immediate early, early, and late. The immediate early genes consist in the essential ICP4, ICP27, and the non-essential for replication 0 and 22. So activation of the immediate early starts with the entry of the viral particle, and the product of this immediate early genes activates the early genes. Subsequently, the immediate early and early genes and viral replication is needed to activate the late genes. So as you can see, deletion of the immediate early genes completely stops virus capability to replicate. So these vectors are incapable of replicating in the host cell and can just be produced in a complementing line. So here I'm showing on the top one of the first vector created, JDNI5. Is a replication defective vector, and it has deleted ICP0, ICP27, the joint region allowing for a payload capacity of about 40 kb. ICP22 is converted to early expression kinetics, and ICP4 is deleted and substituted with a reporter gene. In this case, is a ubiquitin C promoter driving mCherry. We tested this vector for safety, and we transduced human dermal fibroblasts. As you can see here in the graph on the left, HSV wild type cause a rapid decline of cell viability, while our JDNI5 shows no sign of toxicity. Unfortunately, what we noticed was that the transgene payload was rapidly silenced, especially when we removed the ICP0 protein. So, how do we rescue transgene expression in the absence of ICP0? Well, in 2006 was the first time that HSV viral insulator has been identified. There are 7 of them, and they are located surrounding genes that are necessary for the virus to go in latency, reactivate from latency, and other features such as retrograde transport. These non-coding DNA elements are known to be bound by the host CTCF protein. CTCF is a master regulator of epigenetic structure. It recruits transcriptional factors, histone modification. So the binding of these elements by this protein allows expression of the flanked gene. So we wanted to investigate how these elements if these elements will protect our transgene. So we created a vector called JDNI7-GFP, here on the top in A, and we inserted a transgene cassette. This time is a CAG promoter driving GFP reporter gene, and we flanked this by the viral insulator elements located in the latency-associated transcript. These are called CTRL1 and LATP2 upstream from the promoter, and CTRL2 downstream from the poly-A. We created a second vector, JDNI6, that carries the same transgene cassette, this time not flanked by the viral insulator in the LAT locus, but just located in an intergenic region between UL3 and UL4 genes. And we transduced the human dermal fibroblast and looked for transgene expression. As you can see in B, the vector JDNI6, which does not have the viral insulator, does not show a robust transgene expression, while on the bottom, the JDNI7-GFP shows a highly expressed transgene. A second reporter gene located in the ICP4 locus is not expressed in this cell line. In C, I'm also showing that we don't only have achieved robust transgene expression, but also prolonged and stable transgene expression. Now, this human dermal fibroblast expressed our reporter gene up to 28 days. So we wondered, and we wanted to understand what's the relative contribution of this insulator element at this location. So we created a series of mutants of JDNI7-GFP in which we removed the upstream elements, the downstream elements, both of them, the enhancer, all of them. And again, we transduced human dermal fibroblast. We observed that the highest level of expression is achieved when all the elements are present, but we also noticed that the deletion of the upstream elements, CTRL1, rapidly caused transgene silencing. Lesser influence in transgene protection has the downstream element. The enhancer shows high protection since deletion of it caused also transgene silencing. And of course, the deletion of all of them does not allow for transgene expression. So then we wanted to see also if these elements are bound to the LAT locus, or if we can move them in other region of the HSV genome and still achieve robust transgene expression. So we created two more vectors, one called GDNI9. GDNI9 has a transgene cassette located in an intergenic region between 45-46, and GDNI10 has the same transgene cassette located between UL50-51. Now, this cassette was either surrounded by the LAT insulator elements or not. We transduced, again, human dermal fibroblasts, as you can see in B and C, independently of where in the genome this cassette is located. As long it is surrounded by the viral insulator, we still observe robust transgene expression. So now that we accumulate this knowledge, what other cell can we apply this vector to? So we tested a series of cells. So from left to right, we tested foreskin fibroblasts, keratinocytes, muscle stem cells, pre-adipocytes, hepatocytes, and dorsal root ganglion. Again, we transduced them with these two vectors, and we observed that we were able to observe transgene expression from all of these cells when the transgene is flanked by this viral insulator. And interestingly, we noticed that dorsal root ganglion showed also expression of the second reporter gene located in the ICP4. So to avoid confusion about the promoter selected and the transgene selected, we created at this point one unique cassette shown in C, where a CAG promoter is driving this time ZS green and firefly luciferase reporter gene. And we placed this cassette in the LAT locus, surrounded by the viral insulator in this locus, or in the ICP4 locus, also surrounded by the viral insulator specific of the ICP4 locus. And we picked two tissue this time to understand how this will affect the profile of expression. And we picked a less canonical target, muscle, and a canonical target for HSV that are neurons. And upon transduction of this of differentiated myotubes, we observed that under the same promoter and with the same reporter gene, the LAT locus, or better, the elements, the viral insulator elements surrounding the LAT locus, were able to protect the transgene cassette to a higher extent compared to the ICP4 locus. Here we have a luciferase quantification in the graph, and every time point, with the exception of the first one, shows significantly higher level of expression from the LAT locus. What happens with the neurons is similar. We observe the transgene is expressed from both loci, for and LAT, but that in this case, the LAT, the 4 locus, at least in the first time points, showed a significant higher level of expression than the LAT locus, and shows this tendency to be higher over the time course of the study. So if we deliver these two vectors in vivo, will this change the expression profile? So this slide shows the delivery of these two vectors in the hind limb muscle of bulb mice. In A, we have we follow this animal by in vivo imaging system to quantify luciferase expression over a time course. In this case, I am reporting in the upper panel a representative picture of mice at 1 day post-infection, and on the bottom a year later. And in B, there is the entire expression profile of these vectors. The expression profile is similar for the two vectors. It peaks at 1 day, declines at 1 month, and then comes back up and stays steadily high for a year. Now again, the LAT logos, or better, the insulator in the LAT logos, protect the transgene better than the elements surrounding the ICP4 logos. So we were able to reproduce what we observed in vitro. We wanted also to understand what was the main cell type that was expressing the transgene. So a year later, these animals were used to analyze what type of cells was expressed the transgene. So from left to right, we stained muscle section for nuclei in blue, dystrophin to identify muscle cells in red, ZS green, that is the reporter gene coming from the virus, and the merge panel. So the top panel here in blue shows the expression level from the JDNI8 carrying the transgene cassette in the LAT location, and on the bottom in the ICP4. You can observe that the ZS green is robustly expressed from the LAT and to a lesser extent from the ICP4 logos, but in both occasions, the transgene is located in myotubes, is expressed in myotubes that are surrounded by dystrophin. We then evaluate the expression in vivo, this time in the central nervous system. And again, we delivered these 2 vectors in the hippocampus of these mice, followed them by in vivo imaging system. Here in A, again, we have a representative picture of these mice at 3 days and 5 months later. In B, we have the expression profile for the luciferase, and we can notice that again, the ICP4 in red shows a higher expression profile than the LAT. Unfortunately, this expression profile declines by 1 month, even if it stays higher than the background, represented by the black line. We also wanted, again, to identify what cells were expressing the transgene. And so we stained the section for DAPI in blue, neurotrace in red, that is a neuronal marker, ZS green, again, the reporter gene coming from the virus, and the merge panel. We still can observe ZS green positive cells, and this the reporter gene is mainly expressed in neuronal cells since it's co-localizing with the neurotrace staining. So since we observed this decline, we decided to investigate cellular insulator elements. So if the viral insulator elements can protect transgene, we assumed that the addition of cellular insulator elements would influence the expression profile of these vectors. So we screened the literature and found out other cellular elements that were used in other contexts or in other vectors. One of them is a matrix attachment region that showed protection of transgene and tRNAs, transferred RNAs, that also showed protection of a transgene when a repressor was placed upstream from the promoter. So we created a new cassette here in C on the bottom, taking advantage of these cellular elements, and we moved this cassette again, either in the LAT or ICP4 logos, and compared it to the previous cassette where we had just viral insulator. So we transduce again the differentiated myotubes, and if you remember from before, we have this knowledge that the viral insulator in the LAT logos expressed were able to protect the transgene better than the viral insulator in the ICP4 logos. But by adding these new additional cellular elements, we observe that now the ICP4 logos is able to express the transgene. So we have changed the expression profile, recovering a new locus for transgene expression in muscle. What happens with the neurons? So the addition of the cellular elements augment transgene expression, as you can notice in the graph in green and purple. And there was no significant difference observed between the two locations at this point. So we decided to test this in vivo in the central nervous system. We picked just a vector who displayed the transgene in the ICP4 logos and delivered them in vivo. Again, we delivered them in the hippocampus of these mice, and here in the graph, you can see the red line is the virus containing only the viral insulator in the ICP4 logos, and who shows a peak at 3 days and a decline by 1 month. But our new vector with the cellular insulator elements reported in purple showed a higher expression level, a significantly higher compared to the viral insulator on their own, and it stayed significantly higher for the entire time course analyzed. So we analyzed again what cells were expressed the transgene. On the top we have the viral insulator alone, and we stained for Zia's green, and we noticed that by 6 months we have a few cells still expressing the transgene, while with the cellular insulator we still have neurons expressing the transgene, but to a higher level. So now that we have this knowledge, where can we apply these vectors to? Now we can take advantage of the large payload capacity and introducing them big cDNAs. For example, the full-length dystrophin cDNA is about 11 kb. We were able to deliver this and to express it in a dystrophic muscle cell line. As you can see, the vector was able to express dystrophin and robustly also in vitro. Another example is the leucine-rich repeat kinase 2, that is the major genetic cause for familiar Parkinson. We were able again to put a large cDNA of 10.5 kb into this vector and shows its expression in vitro. So to summarize, we achieved to develop a completely devoid of virus gene expression vector without cytotoxicity and inflammation. The payload is about 30-35 kb. Deletion of the ICP0 protein eliminated vector toxicity, but resulted in silencing of the transgene payload. And we were able to overcome this silencing by positioning the transgene next to viral and cellular insulators at the LAD or ICP4 logos, achieving durable expression in muscle and neurons. So I would like to acknowledge some of the people who have worked on this project, especially Dr. Joseph Glorioso, Justus Cohen, and Dr. Yoshida Miyagawa. Thank you very much.